saw me before, right? Attending many of your classes. So I'd like to introduce myself a little bit. Maybe Dr. Abraham introduced me later, but I need to introduce myself more. So my name is Ina Yusuf. I'm a fresh graduate of PhD, like 1 year, not very fresh. I graduated August 2019, 1 year. And I got my PhD in a joint program between US and Egypt. I'm Egypt. And part of my PhD I done it here at Longquest and the Biomed Institute at Dr. Abraham's lab. And part I did in here, so it was like joint program. Now I am a lecturer at the division of Department of Biotechnology and Life Sciences, Bani Suef University in Egypt. And here I am a fellow, like postdoctoral fellow for 1 year, doing some of 1 year or more to do part of starting my postdoctoral career over there in the same lab at the university. Okay, so today we're gonna talk about the cross-kingdom immunotherapeutic target for gram-negative bacterial infection. It seems like very weird, right? So cross-kingdom immunotherapy, we do immunotherapy for gram-negative bacteria, but it is cross-kingdom. So before with Dr. Singh, you have like brief introduction about the vaccine, and usually the primary is the active vaccination, which is how to choose the target, right? The target for your vaccine or for your immunotherapy. So here, our target, it will be like cross-kingdom reactivity, and we will talk about this during my slides, during my presentation. So firstly, as you see, I will talk about the gram-negative, right? So we'll show some data about what is gram-negative and how serious it is, so we can just produce immunotherapeutic drug or treatment for it. So gram-negative bacteria is nowadays like multi-drug resistance bacteria. This is why we are focused on it. Gram-negative bacteria, it was previously easy to be cured, very easy with antibiotic, like regular antibiotic or new antibiotic, but nowadays most of the species of the gram-negative bacteria are multi-drug resistant, especially Acinetobacter baumannii and Klebsiella pneumoniae. These two kinds of bugs, they belongs to gram-negative bacteria, and they are almost resistant to most of the antibiotics. Especially Klebsiella pneumoniae, it is resistant to the imipenims or carbapenem class of antibiotic, and it makes it aggressive in its infection. Also, it is hard to be cured. This makes the high mortality of this gram-negative bacteria because there is no treatment, especially for the bloodstream infections. It is good to know that these kind of bugs, it infect mainly the immunocompromised patients or people. Another immunotherapeutic treatment or drugs. This is after the revolutions that happened to this industry, because it is, as we will talk later. So the lecture here, it will be divided, I forgot to tell you, it will be divided in 2 parts. First part, I will talk about this project. And the second part, we will talk more about the monoclonal antibody production and technology, and what is happening on in this industry and investments. So we will talk later about this technology, but it's good to know that this technology is growing day by day, and it's very, I couldn't say very easy, but it's like very visible to yeah, what is it? Yeah, okay. Yeah. So instead of having the antigen of this bug or this pathogen, we use the antigen of other pathogen to introduce immunity against this pathogen. So simply, if I have like pathogen X have antigen on each surface, I use this antigen to get immunity against antigen against pathogen which is Y. These 2 pathogens could be on the same kingdom, like bacteria and bacteria, so I can use antigen bacteria for E. coli, for example, to treat or to vaccinate or to produce immunity against another bacteria, which could be Salmonella, for example, or whatever. So this is called within the same kingdom. Both are bacteria. Or we can just having antigen from fungi, like Candida, for example, that we can produce immunity against a bacteria, like gram-negative bacteria, and this is what we have here, right? So what we have here is that we will have like a candidal antigen, that I will talk later about it more, and produce immunity against bacteria. Very weird. So how this come? So why a different antigen from different kingdom can have like this cross reactivity to this bacteria? Do anyone have like any ideas? How this, if for example the Candida antigen, when you produce antibodies against this antigen, it can protect the infection from bacteria. How this happen? How this antibody recognize the bacteria which is don't have the same antigens as Candida?
Homology.
Perfect. Structural homology, right? So there's many factors affecting this. Homology, protein structure, homology. So both proteins are having some sort of homology or similarity between them, either 3D structural or even on sequence based. Or also similar host pathogen relationship, as we will see in our case. So both pathogens have the same tissues that they invade, or the way they invade, or maybe they have the same feature of virulence. So they have quite similar, the same, we can say, character of invasion or pathogenesis, the same. So we can expect the same this protein to have the same between these two both pathogens. Okay, so is this clear how this produced? Okay, so our story starts when I was not here at that time. Okay, it's like 8 years ago. But the story starts 8 years ago or more, when people was publishing that Acinetobacter baumannii bacteria interact with Candida albicans in the catheter and can just help both can synergy their effects in invading or infecting the catheters, its heart or whatever the catheter where it is. So they have like synergistic effect both together, and they interact with each other. So Acinetobacter baumannii interact with the Candida albicans, and Candida albicans help Acinetobacter baumannii to invade the cells. So they are in a mutualistic relationship. Then they thought about what interactions this happen through. There's a lot of experiments done to know how these two organisms interact with each other. In other words, what is the protein here interact in what's protein here, right? Because we are talking about microorganisms. So when you are talking about microorganisms, so any kind of interaction will be protein-protein interaction mostly, right? So the OMP-A protein, which is outer membrane protein class A, in Acinetobacter baumannii interact with HYR1 with hyperregulated protein number 1. And this kind of interactions help both of them to survive and infect catheters especially. Okay, so yeah, this hyperregulated. Also, it's good to know that HYR1 is important it helps the Candida albicans to resist the phagocytes and help bacterial adhesion as well. So it helps, this HYR1 helps Acinetobacter to adhere more, and also the biofilm formation. So the idea came, poop up, yeah, so there is a homology, maybe there is a homology between some of the proteins of the HYR1 and or the Candida and the Acinetobacter baumannii. So thanks for the bioinformatics tools and tech tools that help us to do a lot of modeling and bioinformatics tool in silico, like on my laptop, that it can compare between the 3D and predict also. Compare and predict the 3D structure of the protein and then compare it with other proteins. So what Dr. Mike Heeman did, he just do this way. And what he have here is that the HYR1 3D structure of HYR1, it shows that it is similar, and you can see it is similar in structure to another protein in the Acinetobacter, which is called filamentous hemagglutinin adhesion. So the structure of HYR1 in Candida is similar in the structure of FHAP or filamentous hemagglutinin adhesion in Acinetobacter. And we don't also concern about the structural homology. We're concerned about a lot of things. So is this protein is outer membrane? So what do you think? Filamentous hemagglutinin adhesion, right? So it is outer protein. Because if there is a homology between the outer protein of Candida and inner protein or intracellular protein, what is the point? I need outer membrane or extracellular or whatever outside to be, targeted. So it is outer protein. So we are lucky. So it is outer protein, and also it is also important to be important in virulence or pathogenesis. And you can see this helps the bacteria to adhere more. So it's help in the adhesion. So this is also a virulence protein. So they got interested in this also with OMPA, but OMPA came later. You couldn't see a huge homology, like very short part here, this part which is homology with this part, and then in sheet part is the N-terminus of HYR1. So there is more than one protein in Acinetobacter that we can say this term cross-reacted with the HYR1. So they have like some sort of homology. So we can then apply what is called convergent immunity, right? So this is what we did. First, we started to do some in vitro. They do, because I didn't use that. So they do some in vitro assays to see whether this would be applicable or not. So what they did, they took this recombinant HYR1 protein or antigen, and then they produced antibodies against this HYR1 protein, and then they checked the antibody capability of binding to the surface of gram-negative bacteria, primarily Acinetobacter, because this is the first bug that we will talk about, right? So they put Acinetobacter cells, and they put on it the antibodies against HYR1 and see if these antibodies will recognize any receptors or any proteins on the surface of Acinetobacter or not. And they also introduced another organism like a control, which is Pseudomonas aeruginosa. And what they say, they use flow cytometry to do that. So after the binding, so this is, if we can say that this is the cell, the bacterial cell, and the antibody recognizes something here. So the antibody binds. So how I can know that the antibody binds or not? I just use secondary antibody, that is fluorochrome, and then use flow cytometry, for example, to detect these signals. Okay? So this is a flow cytometry chart histogram. You can see here's the control, control antibody, which is isotype, for example. And this is 2 doses of the anti-HYR1 antibodies. You can see here there is the events, all the events will be in the positive way, which is all fluorescent, by 94% or 69 at lower dose, compared to the Pseudomonas aeruginosa, which didn't show any here binding of this. So this encouraged us to do pilot experiment and see what is this active immunization. Active immunization means what?
That your body starts producing.
Yeah, so what I'm gonna do, I will just get the body with the antigen, and the body by itself, it will generate the antibodies, and after 1 or 2 boosting, I will give the infection or challenge the person, but it's not person, it's mice, okay? So we just do active immunization. We prepare recombinant protein of the HYR1 and then inject either way, IP, or you have this with Dr. Singh, right? That there is different ways to get the antigen to the body, and yeah, it's a very big start. So they just immunize the mice. We here use mice or mouse models with, so we immunize the mice with the HYR1 and boost once after 21 days, and then after 14 days we infect. Infect with what? With what? Acinetobacter, right? Oh, you are here? Okay. So we infect with Acinetobacter. So we expect to have some antibodies, so the body produce some antibodies against the HY1, which is could cross-react with Acinetobacter and help in survival. So we can see here the survival curve when we compare it with the alum, which is the adjuvant only. With the here is the alum, and here is the active immunization mice, which is immunized with HY1. So there is significant difference between both of them in the survival mean. This means that the HY1 produced antibody against HY1, which was protective against the lethal dose of Acinetobacter in mouse model, right? This means that. And this means that the conversion immunity happened, right? Is this clear?
Yeah.
Okay. Also, we test this in the kidneys to see the dissemination. It happened in controlling the controlling the bug from dispersion. So you can see here the alum versus vaccinated mice. You can see there's a bacterial burden in kidney and lung and spleen. There is significant difference between both of them. This means this immunization helped in curing the mice that is infected with Acinetobacter baumannii. Okay? So further, as we said, we were talking mostly of this project will produce passive vaccination, which is mean antibodies. So what they did, they took the HY1 is like big structure. So they take part of the N-determinants and having some with the in silico, also with the in silico tools, they having choose like candidates like 15 amino acids only, peptides, candidates for the to be like a vaccine. So in this previously we took the whole protein as a for vaccination, right? Here they trying to produce antibodies against the protein. So they didn't take all the protein. They took like 8 peptides from this protein. Depending on what they took this, they chose this peptide. It is in silico analysis, depending on the T epitope, B epitope, and that they are will be more immunogenic. So they pick up 8 peptides from the HY1 and they name 1, 2, 3, 4, 5, 6, that's 8. Okay, and then they test these antibodies as a protective agent against Acinetobacter lethal infection. And again, you can see like compared to the alum, or sorry, compared to the placebo, which is given like isotype antibody. Do you know what is that? When I say isotype antibody means antibody which is the same isotype as our antibody of interest, but without any recognition, specific recognition. So these antibodies like didn't recognize anything, okay? You know, like theoretically, they didn't recognize anything specifically. So we use this as our placebo group, and we infect the mice with the Acinetobacter and see what is the difference. And you can see here is the peptide number 5, which shows like more than 90% protection. Okay, so we pick this peptide to do further studies on it based on the survival study. So please let me know if you something unclear or you don't understand anything or just something dropped with you, so we can follow me properly. Okay, so till this point, is this clear? Yeah, please.
So when the mice are infected, are you injecting them immediately with the antibody for passive immunization?
Yeah, okay. Yeah, this is a good question. So here, usually the vaccines, it happens you have the antibodies before infection, not after, right? Mm-hmm. But in our case, yeah, we treat the mice after infection at day +1, which is after infection by 1 day.
Okay.
So you can say this is like treatment more than prophylaxis. Mm-hmm. So we are challenging this HY1 antibody to see if they are just protect like antibiotics, because you take antibiotics after infection, right? Mm-hmm. So we are treating them in this way. So if it is like protective after infection, it will be more protective when you take prophylaxis, right? So we challenged ourselves more. We tested this in 2 models. So when Dr. Mark talk about the models and how we choose a model and this stuff. In our case, we have a mice model which is lethal. I said lethal. This means that because our model is to the mice all or none, means if they die or survive. So we have a survival model. Survival models means that the mice should die. So I infect them with a lethal dose, means very, very high dose of the Acinetobacter or whatever bug. And usually most of Dr. Ibrahim's lab using survival models, okay? So the mice should die, so I can see death, okay? So we infect with a high dose. And these models that we use is either diabetic or neutropenic. So both of them. So do you know why we didn't use like immunocompetent, like healthy mice? We use either neutropenic or diabetic mice. Do you know why? Sorry?
So they wouldn't have like an active immune response against your pathogen or Acinetobacter.
And this how could help in our experiment.
So now you can see if your experiment works, I guess.
Oh yeah, so you mean that you suppress the immunity, so only the antibody, our antibody will work only. This is your point? Yeah, this is good point, yeah. So what else?
We'll have the population that is actually having this disease are immunocompromised.
Yeah, perfect. So we said this also that these bugs infect mainly what? The immunocompromised, immunosuppressed patients that they are hospitalized or have injury or whatever. So we need to make it like compliance, like the same with the real, and also to make the antibody see if there is no immunity or less immunity, what we're gonna have, right? So we choose these 2 models, diabetic and neutropenic. Also diabetic because usually the wound infections and this kind of stuff, it happens mainly for diabetic, right? So this kind of could be like resemble the real population that will be infected with this bug. Okay, so also in another model, which is neutropenic model, we see like huge difference between the placebo and the treated mice with the anti-HY1 antibodies. We tested here different doses. So interestingly here, yeah, we have a very encouraging in vitro and in vivo study. But the main question is how? How this HY1 cross-react with that, right? We need to know. Because otherwise you will be like, maybe this is like not real, or this is, yeah, we can see it in the lab and in vivo and in vitro, but we need to know exactly how these monoclonal antibodies against HY1 be protective against bacteria. So we did Western blot. I think this is mine. We did Western blot to see what outer membrane proteins on the Acinetobacter interact with HY1. Do anyone here did Western blot before? Yeah, so do you know the idea of Western blot, right? Mm-hmm. So what we did here, that we just run on a gel, SDS page gel page, the outer membrane protein of the Acinetobacter, okay? And then we transfer that protein to a membrane, okay? And this membrane having this protein in the same position, and we have the marker for that, okay? And then we took this membrane and then block it and do several steps, and then put the anti-HY1 antibodies that we have, and then we visualize it by whatever, fluorescent or by imaging or X-ray or whatever. So if you see like a band, this means that the antibody binds to that band, to that protein. So it binds here, we can detect here that it binds to 4 bands. So we can see 4 bands. This means that this antibody recognized 4 different proteins. they are different because they have like different possession. We here did 2D Western blots, that is depend on the charge and the size. Okay, so we just separate the SDS page is to separate the proteins over the gel, whatever 1D or 2D, and then put your primary antibody, which is anti-HYR1, and then visualize it by either way, fluorescent or X-ray, and you see this band. So I know this band, but I don't know what is this protein corresponding to, right? So we just compare the membrane with the gel and see, yeah, here's the band here, so yeah, and this is corresponding to this band. So I cut this band and then send it for sequencing, so I know the sequence of this protein, and then I can recognize it. So is this clear? Clear? Okay. So generally, so we just know this protein that is cross-reacted with HY1. This means that OMP interact with the anti-HY1 and TOMB dependent and other proteins, which is also FHAP. So now we confirmed the in silico data that we have, right? In our computers, we checked that HY1 and FHAP cross-reacted. Now we just confirmed this with practical experiment. Also, we found another proteins like OMP, ultramembrane protein, and TOMB dependent. TOMB dependent, it is a receptor for the iron uptake. So it is also virulent. So the most important thing of these proteins is they are outer membrane and they are help in virulence as well. Okay? Yeah. After this, they said, yeah, maybe this HY1 have a broader activity, like it will be kind of cross-react with other organisms, right? Like Klebsiella pneumonia, which we are interested in. So they did the same, exactly the same. They do some homology, computational homology between the Candida HY1 and KP, and they found also there is virulent outer membrane protein, like the same as OMP and TOMB, and also another protein for fembrile adhesion protein for the femprii. Do you know the femprii rule is, right? The femprii is something like protruded outside the bacteria that help it moving this stuff. So yeah. So this is this is also virulence factor. So we did the same. We checked the binding also with the antibodies. And at this step, we use the monoclonal antibodies generated, which is IgM isotype, and we tested against HMC1 is our isolate clinical isolate of Acinetobacter. Here, we are using all the isolates that we are using are clinical isolates from the hospital. They give it to us, we characterize it, and we use this for our experiments. Because we need our experiments to be like, resemble the real. So the same pathogens that is emerged in the hospital is the same that we are working on. We are not working on the APTC strains or like standard strains. We're working on the clinical isolate strains, which is multi-drug resistant, right? And this is our target. That our strategy of treatment is like our experiment design will be the same as what will happen in the real. So we took this. All of them are multi-drug resistance clinical isolates. Okay. So H1C1, which is Acinetobacter, you can see binding. Also, we did the same. We did flow cytometry to check if they are binding or not compared to the isotype. So here you see the isotype, and you can see here there is a shaft in the bands. Means when there is a shaft over here, this means that more events fluorescent. So this means that there is more antibody recognize the surface of these bags. This happened for HMC1, Klebsiella 2 strains. We have carbapenem resistant Klebsiella and multi-drug resistant Klebsiella, but it's not carbapenem. We tested both, and they have like very nice shaft in the peaks, which mean good binding. Also, we tested lower concentration to see, and you can see here, like at lower concentration, there is still binding of this kind of all these 3 bugs. So now we have 2, we are counting now, we have 2 organisms, right? And that is reactive against, means H1 reactive against them. other isolates. We tested HMC 6 and 12. These numbers is like serialing when they have these clinical isolates from patient, like they name it 1, 2, like that. So, and also there's the KPC, there's different isolates. So when we test these in different isolates, we would trying to know whether it is like because of this isolate, because you know the clinical isolates not the same as the ATC. They have a lot of genetic modification on that bacteria, right? This is because it is multi-drug resistant. So it is not like the wild type, not wild type, the ATTC strain, or the strain which is not drug resistant. So we want to know if this is like specific for this isolate or it is like general for all isolates. We can use it like generally. And then we get like the same shaft in the answer. Yeah, it's not isolate specific. Also, we do some in vitro assays that show up if there is any protection from damage or not. So we challenged mammalian cells, epidermal cells, or endothelial cells with, we challenged it with bacteria, as Acinetobacter or Klebsiella pneumonia, and then firstly we incubate the cells, the mammalian cells, with the antibody. Okay? We have our cells, okay, mammalian cells, and then we incubate our bacteria with antibody. So you know one of the mechanisms of the antibodies that it neutralizes the virulence factor of the bacteria, right? So we incubate them together 1 hour, and then just challenge the cells with challenges the cells with these bacteria that is having the antibodies, and see if there is protection from damage or not. We expect when the cells, mammalian cells, attach it to the bacteria, well, it will be damaged, right? So, and we check compared to the isotype. You can see here, this is a percentage damage. So there is a decrease in damage. So this is a percentage of damage. So when it is less, so this is a reduction in damage in 2 of the clones that we have the antibody. This is repeated in the, also for the endothelial cells and the epidermal cells. Different concentration. Yeah, this was very encouraging to us because, like, you can see here in some clones, it totally protects, right, the damage that happened to the cells. Is this clear for you? How we designed this experiment? Okay. Okay. Also, we tested an animal model in the mice these antibodies against both Acinetobacter and the Klebsiella. We tried to repeat this again because we have new monoclonal antibodies this time. Previously we have polyclonal, now we have monoclonal, so we need to repeat some experiments to see that everything is concise and like repetitive. Okay, so we did this the same, and you can see like, yeah, very protective, like 100% or like 70%, and you can see in the lung there is significant difference between the treated one and the placebo. Also the same, you can see from the Acinetobacter, from the Klebsiella, you can see the like very significant difference between the placebo and the treated, also in the lung. And the same, we tried to know what is the protein that's cross-reacted was the same. So now we confirmed with it's important to confirm your in silico or your bioinformatics data with the real experiment as soon as it is possible. Okay, so we confirm it also the same, and then yeah. So the next step. So the second half of this part, this is what part of my PhD. So during my PhD, I did that work that I showed before about the monoclonal and the Klebsiella. I did the whole Klebsiella cross-reactivity and this stuff because I did Acinetobacter before I came. And then now I'm continuing working on the same project. I don't know if I'm lucky or not, but I asked other people. Yeah, he said, I didn't open my thesis after I done. I said I open my thesis every day because I work in the same, right? Yeah. Okay, let's say I'm lucky. Okay. Because yeah, why I didn't do that during my PhD? Yeah, why I didn't write this? Because you are grown up, you are, you learn more, so you criticize yourself. Yeah, why I did it? Yeah, well, no, it's like, that's okay. It's good to have self-criticism. So here, this is what we're gonna do next step in that part. So we will raise IgG isotype because it is more convenient to have IgG. Previously, we was working with IgM. IgM has a lot of issues, like they are less stable, short half-life, big structure. It is pentamer, if you remember. So most of the people use either IgG or IgA. So do you remember IgA, where it is? The mucus, yeah. So if your microorganism is coming from oral or it will contact with the mucus, it's better to have IgA. It is like a couple of therapies in markets that have IgA. But usually people like have IgG, which is systemic, so it has like this systemic effect. So we are trying to raise IgG right now. We are in that process. We are not raising it here. For the IgM isotype, I did it in my PhD. So I raised the IgM antibodies, and I will talk about this later. It's a long story. But here we give it to company to do it for us, okay? Because it will be faster and it's a lot of work and need a lot of labor work, and it's like nonsense to just waste time. It's just give it to company and give it to you, and you do your experiment. So then we will, I think we are in that part, so are reducing mutations. So what we're going to do in this part, we need to know if that result that we see, it is confirmed or not. So how are we going to do that? So we do knockout for the bacteria, either Acetobacter or the Klebsiella, knockout of the targeted protein, okay? And then we'll see if there is effect of the binding of the antibodies with this mutant based on they are cross-reacted in this protein or not. So now we'll confirm that the protective activity of the antibodies against HY1 is through that kind of protein. This is like confirmatory of the, because if you see a lot of papers, I think most of you like have original paper presentation, right? So in original paper or original research, they do a lot of experiment just to confirm their idea, because like 1% could be these results, like not the way you explain it. I explained this Western and about that in this way, but maybe it's like not. So usually people do that, like do knockout of these proteins and then check. So we are done with this, and also this part, big part done by Dr. Bahgat. He was postdoc at the Dr. Ibrahim's lab before I came, and I will continue on the same mutants to do a lot of work. So we will investigate and characterize the pathogenesis potential of these generated mutants. We will check these mutants if they affect in their variants, because the protein that we will knock out is virulent, right? So we need to see if the mutant will have different variants from the wild type or not. I mean the original isolate, right? And then we'll see in vivo studies the infectivity of this, and then what is very interesting that we will generate, if we confirm it with that, this part, we'll generate recombinant proteins from the Acetobacter like OMP-A and this, and try to vaccinate Candida. Alright? The vice versa, like all the way around. So we'll just see if this kind will have a protective or not. Lastly, I'd like to acknowledge my team, which I'm very proud of. Dr. Ashraf and Priya and Dr. Jack Shore, the mentors, our Dr. Ibrahim of the lab, and also other people that helped in that project. This is a big project, huge project, so I didn't do all this by myself. We have several people that worked on it. Also, I'd like to thank NIH for the funding, and also Fulbright and my country, Egypt, because they funded us. I forgot to tell you that I'm a Fulbrighter. I don't know if you know Fulbright or not. No? Okay. So it seems all of you are American, right? So yeah, so you don't know about it. So Fulbright, it is an agency that give funding for students, masters or PhD to pursue their research or their masters abroad, like especially not abroad, in the US, okay? So it is a US organization affiliated to the US government, state government, federal, and they funded me during my PhD to come here to do part of my research. So this is why I forgot. I don't know why I forgot this. That's okay. So now, yeah, we are done with the first part. I don't know if you're running time or not. Yeah, we are not. We are good. So we'll have break like 10 minutes, and then we will come back and talk more about the monoclonal antibody production and technology, and yeah, try to get some coffee because I feel some of you also like. Okay, sounds good.